Good morning, good afternoon, and good evening, everyone. Welcome to my stream. Today, we are talking about a drug. The process of drug development and discovery are time-consuming and expensive with high failure rate. Clinical failure rate from phase 1 to drug launch is over 90% constant. And the cost of research and development double almost every 9 years. There's pricing demand for using machine learning and deep learning to help improving the drug research and the develop pipeline. However, the million-dollar question lies, can the machine learning really speed up this ancient field of searching and developing drugs? Dive a little bit deeper, machine learning and deep learning algorithms, performance, and outcome are heavily depend on the data quality, especially labeled data, which is traditionally manually labeled. In drug discovery and development, the process labeling data is extremely labor intense, expert requiring, and cost inefficient. Another important question would be, where are and what kind of data can we utilize? I'm Bill Chen, stream owner, and currently work as a machine learning engineer at the San Francisco Bay Area. Welcome to ACE Machine Learning in Biology. ACE is a community of machine learning practitioners and researchers who would love to gather together and talk about the topics in AI research, engineering, and products. We host free live sessions like this 3 to 5 times a week and produce high quality content in various subject areas. To see more about this, go into the AI science, log in and access slides from this and other sessions and many more. Also, make sure you subscribe our YouTube channel, Machine Learning Explained, to get notified about all the live sessions and other free content we publish. We currently have more than 30 different streams that are focused on machine learning topics. And this session is in Machine Learning in Biology stream. Hope you enjoy it and coming back. Today, we invited Professor Dr. Sally Ellingson, and her topic is data exploration. Can machine learning discover new drugs? Dr. Ellingson is an assistant professor of the Division of Biomedical Informatics, College of Medicine, University of Kentucky. She is a computational scientist working at the intersection of computational biology and high-performance computing. She has an undergraduate degree in computer science and mathematics from Florida Institute of Technology. She obtained her doctorate degree at the University of Tennessee and the Oak Ridge National Laboratory under a fellowship funded by the National Science Foundation in computational biology. In her additional role as manager of high-performance computing service for the Markey Cancer Center's Cancer Research Informatics Shared Resource Facility, she facilitates high-throughput genomics and big data processing for precision medicine, resulting in targeted cancer therapies. Recently, she has been combining simulation, big data, and machine learning to increase the accuracy of drug binding prediction. With her passion for high-performance computing, her research goals lies in harnessing computational power for discoveries otherwise not possible in biomedical area of high social importance. High social importance. Without further ado, let's start our today's talk on data exploration. Can machine learning really discover new drugs?
Thank you very much for the introduction, Bill, and thank you for inviting me to give a talk on your YouTube stream. So first of all, what is the motivation for using data and machine learning methods in drug discovery? It's very expensive and hard. That's, something that we hear often in this field, and always these talks start with, the general, it's super expensive. It costs a lot of money, as Bill already brought up. So some recent studies showed that now they're looking at the median cost of bringing a drug to market to be about $985 million. This other recent study so that they can go up to about $3 billion, and only about 12% of the drugs in clinical development reach the market. And because of this high failure rate, it additionally adds to the cost of the overall R&D for drug development. And there's lots of stages in drug development shown here. Step one, discovery and development, that has to do with, finding out what the basis of the disease is and drugs that target it. And after that, things go for animal testing to better understand it. And then after that, there's clinical research to make sure that it's safe and effective. And then there's still more review to be done by the FDA and post-market safety monitoring. And all of these steps are very lengthy and costly, and often they fail late in the phase, which, is part of the reason that the cost is so high. And so the focus here and what my research is and computational methods that I do are in the initial stage. So in this initial stage, you do things like identify the molecular basis of a disease, what's causing it. Also, finding a specific molecule, one of the causes of the disease, that's able to be targeted by a drug. And then also ensuring that this drug also doesn't negatively affect you because it's binding to different things and different pathways that are important to your survival. So for the first one, these are examples of why this is so hard. So identifying the molecular basis of a disease. In this picture, this is an example of pathways in cancer. It's not just one particular protein, usually that has some defect. Sometimes it's multiple, and these pathways can be, very complex and not completely understood. And even when studying these, and researchers believe they've identified a gene or a protein, that is relevant to the disease, and they are intending to target with the drug, and they see good results as far as, shrinking of tumors and being able to get better from the disease. Many studies have gone and shown that actually these proteins that they're assuming to study to be, responsible for the disease and for the drug to be targeting aren't what's actually happening in the body, because it's very complex what's happening. And then the next one is to find a specific molecule that can be targeted by a drug. So, proteins are tiny molecular machines that perform most of the tasks that keep cells alive. When there's things that are wrong with these proteins, such as they're more than there should be, or less than they should be, or there's mutations in them, these are often causes of disease. And drugs, are tiny small molecules that potentially combine to the proteins and turn them on or off and modify what's happening and causing the disease. And then the next one is ensuring that the drug does not negatively affect other pathways. And so this is also a very complex and hard problem. This picture in the background is a picture of a biochemical pathway chart. So these are all proteins and, things that have upstream and downstream effects to each other. If one protein has some signaling effect down the line, something else could. And these are very, very complex pathways that proteins overlap and are involved in multiple of them, and they're not completely understood. So making sure that we don't negatively affect important pathways is a very complex problem. In addition to that, drugs have been shown to be very promiscuous, meaning that they probably will not bind to just the protein of interest that is relevant to the disease, but it can bind to lots of other things in the body. And this study that did a prediction of a genome-wide drug-protein interaction, they said our predictions show that drugs are quite promiscuous, with the average number of human targets per drug of 329 and the median being 38, while a given protein interacts with 57 drugs. And so the focus of the work that I've done and what I will talk about is on the computational prediction of drug binding. So, given a protein that is relevant to a disease, finding drugs that can bind to them. And so here's an example simplified with shape. So this is the target, that's the protein that's relevant to the disease. Then we have a chemical compound, and we just want to predict how well they fit together. And here shown 3D structures of the protein and also drugs. And this is in reference to a calculation that's called molecular docking that has simplified scoring functions so that they can do quick calculations. And these scoring functions are influenced by the shape, size, and physicochemical properties of both the protein and the drug. So even if we were able to do these predictions very well, to understand the entire, scene of what's happening in the human body with all the proteins and all the potential drugs is a huge problem. So how many human proteoforms are there? So there's just over 20,000 genes that we know of, but genes can encode proteins in different ways. There's splicing and different ways that they can form proteins. And in this paper, they estimate that the number of different proteoforms is in the millions. And then on the right side is an estimate of how many potential drugs there are. So this little graph shows just some relevant sizes to circles of this one is the drug-like chemical space, and over here is the biologically relevant chemical space, and there's estimates of this being up to the size of 10 to the 60. So what is the data and machine learning solution motivation? That's exactly what it is. It's the size of the problem. If we were able to do these calculations and predictions at a big scale, we'd be able to very quickly repurpose drugs, understand the side effects. But how feasible is that? Um, so just a little thought experiment to back up, why having machine learning solutions that could be quickly calculated based off of models would be very usable. So this thought experiment is to make calculations for how much it would cost to experimentally validate the interactome between the drug-like molecule space and the druggable biological space, and then how much time would it take to do computationally. So we showed from the other papers that there's predicted up to 10 to 6 proteoforms, different structures of proteins, and 10 to the 60 small molecules. And then based off of just looking at some assays that are out there, we're using 10 to the third as the cost for an assay. But really, there's many different assays. They have different advantages and disadvantages, and experiments need to be repeated multiple times usually. But assuming we pick the right assay and it works correctly, we could experimentally test all of these for 10 to the 69th dollars. And what is that number? So just for fun, I looked up something that is 10 to the 69. And so it's 10 to the 69 bit is the thermodynamic entropy of the Milky Way galaxy. And entropy is a measure of the system's thermal energy per unit temperature that is unavailable for doing useful work. And this is also a measure of the molecular disorder or randomness of a system. So if the randomness of the Milky Way and molecular disorder was a dollar amount, we could use that to buy the assays to do these experiments. But that doesn't really include, the people power and everything else that we would actually need to do this. So obviously it's not very feasible. And then to do it computationally, in this movie right here, it shows molecular dynamics that was done on a special purpose supercomputer that's designed just to do these calculations so they do them more efficient. But unable to do a simulation long enough to get the kinetics to actually do to predict binding affinity, it's usually takes some time. And in this, they took about 5 days of compute time on a special purpose supercomputer. So if we're assuming it takes 5 days to accurately do one of these calculations, it would take 278 years to look at one drug against all gene products, and it would take nearly 14,000 years to look at one drug against all proteoforms. So again, even computationally, to try to accurately do these predictions at this scale is not possible. So that gives us, why it's so important or could be so useful if we had machine learning methods for it. There are a range of prediction methods that involve simulations with very computational times and accuracies, but none of them are perfect and all of them have their downfalls. So what if we could use the mass amount of biomedical data that we have available to have knowledge discovery and predict new drug and protein complexes? So here's an example of a kind of data set that can be used for this. So this is just a depiction of a drug binding benchmark data set. And then in these data sets, they have, for a particular protein, they have a large library of chemical compounds, and they have some subset of them that are known to bind to the protein, and then some subset that do not bind to the protein or are called decoys often. And then these benchmark data sets usually have it for multiple proteins, sometimes in the hundreds. And then here's underneath this block is some properties of these benchmark data sets that when they were originally made, I don't think that they were thinking about their usefulness in machine learning that could potentially cause problems with using them in machine learning. And we'll look at them after we look at an example of what can happen. So to do machine learning, we need to have a way to featurize our data sets. There's many, many different ways to do this, but just as, an easy example, drugs can be featurized in these bit strings where the bits are on or off depending on if they have different substructures in them. And then proteins can also be characterized in many ways due by their primary sequence, their secondary sequence, and their tertiary or quaternary sequences, as well as, in particular, the binding site. And different things can be calculated, such as the volume, the surface area, and the different types of atomic contacts that are available. So here's a study that took a benchmark data set, and the data set had 102 proteins in it. And as I mentioned, in these benchmark data sets, each protein has a set of stuff that binds to them and a set of stuff that doesn't bind to them. And in this study, they took 72 proteins for the training. So all of their active compounds for those 17 proteins, where those are the positive examples, and the non-binding or decoy compounds were the negative examples. And then they took 30 proteins to test, and they received a very good mean AUC of 0.904. But in this, they only used the features that come from the drugs, which were the fingerprints, like the binary string that I just showed, and didn't have anything at all to do with the actual protein in it. So is there something that is just making these data sets easy to classify that has nothing to do with really predicting whether or not a particular drug will bind to a particular protein? So here are some of the properties that I had blocked off before to talk about after showing that problem. So what happens when these data sets are built is that these decoy sets, the non-binding ones, they're not usually from all experimentally verified. Sometimes they are, but to really mimic a large screen, these data sets were built to have a very large number of decoys, so most of them were generated. And they were generated to be similar to the active ones by using simple features that, like their molecular weight, number of hydrogen bond donors and acceptors, just because they wanted to make them not easy to classify. And then the actives are generally all experimentally verified from things that were actually done in the lab. And when compounds are experimentally tested in the lab, a lot of times very small changes are made to scaffolds in order to test how much better or worse it is. And then what happens is active sets are enriched with very similar things. And then to make sure that there are not things in these generated decoy sets that are actually things that should be in the active set, they usually do some filtering to make sure that anything that is not almost exactly the same is removed. And then with this whole process, they could be making data sets that are easy to classify based off of things that are not relevant to the binding. So here's an example of something that can be used to quantify this. This is spatial statistics, and they were developed when creating a benchmark data set that was trying to alleviate some of these problems. And so for in these examples that I'm going to show, in them, we're creating a vector of features that all have to do with very simple things, mostly the total number of atoms of different types, and then using a simple Euclidean distance as the distance function. So the way that these metrics work is that there's a function I where when there's the distance between the two compounds based off of this feature vector and the Euclidean distance are within some threshold, it's 1. And if it's not, then it's 0. And then there's a nearest neighbor function, which is this g, and this is the active to active distance. So it's the number of actives or the portion of the actives in the data set that are close to each other. So here's an example that comes from a real benchmark data set. This is the name of the protein right here. And so this means this purple line is the this nearest neighbor function. And so this is the distance, 0 to 5, and then 0 to 1 is the portion of that data set that are within a distance. So we can see for on this top data set that close to half of them are very, very close to each other based off of just very similar things. And then the empty space function, f of t, is the decoy to active distance. So this shows the portion of the decoys that are close to an active. And so we can see here on the top example that the f of t stays close to 0 until nearly after a distance of 2, meaning that all of these decoys are very dissimilar to the actives in the data set. And you can see that they never get to be close to each other. So this is an example of a bad data set that's very easy to classify. And then this bottom example is one of the better proteins in the data set based off of these statistics. And you can see that the nearest neighbor and the empty space function closely overlap with each other, meaning that they're very similar in these simple features. And then this other function is a term for data set clumping. And that's a term that quantifies when all of the actives are closely grouped together and easy to classify in terms of the decoys in the data set. And it is a difference of f and t. So then the sum of this S function is the bias score. And when it's near 0, it means there's low bias in the data set. So as you can see, when you sum up this difference over it and it's close to 0, it would mean that these lines very closely interact. And then if you sum up the difference in one like this, you would have a large value, and it would show that there's a lot of bias or data set clumping where the active compounds are easy to classify. And then so using those same statistics, but doing the sums of all of them, a large sum g, the nearest neighbor function, indicates a high self-similarity of all the actives. So all the actives are very close to each other. And then again, with the sum of f, a small value indicates a high degree of separation from the actives. And then the sum s, a negative value indicates clumping. Near 0, as I described, means a random-like distribution, which is what we're looking for. And a positive value indicates a larger active-to-active distance than decoy-to-active, which might show that there's a problem in the data set. And so what's shown here in this graph is the x-axis is each individual protein in the data set. And the sum S is supposed to be near 0 to show low bias, and we can see that there are very few proteins in the data set that actually have this, showing that using these statistics, we can see that there are a lot of problems in this data set as far as making things easy to classify based off of actives being very similar and dissimilar to the decoys. And here's another paper that revises these spatial statistics to include the training and testing aspects of it for particular use in machine learning. And so in these, they have what they call the asymmetric validation embedding, or their AVE bias. It's very similar where these H functions are a cumulative nearest neighbor function as seen before, but in here, they look at the validation data and the training data. And then To shorten it, these terms are called AA minus AI, and that is for active set clumping. And then they also include in here, which wasn't done previously, is inactive set clumping, because while it is a known problem that all the actives can be very similar to each other and making it easy to classify them compared to the decoys, it could also be a problem that the decoys are all very similar to each other, making them easy to separate from the actives. So there's also a term for the inactive set clumping. And in this, they use this to create splits to try to reduce the bias for machine learning. And the goal is to have a B that is close to 0 to have a less biased data set for machine learning. And so here's an example of looking at the same benchmark data set that I just showed. After running this to create splits that are optimized using this score for machine learning. And so what's shown here is the AUC for some standard classification algorithms, and then also the bias score. And some things to note from here is that they use k nearest neighbors with k equals 1 to detect a potential for overfitting. The idea here is that k nearest neighbors with n equals 1 just memorizes the training data because the answer is always whatever the closest thing is in the training. And we see here that we do have a close correlation with the results of k nearest neighbor with the bias score. And then over here, we have the breakdown of the active set and the inactive set bias scores that determine their individual clumping. And so some things to note about this, we can see that the most biased splits, the ones with the highest bias score, have the best AUC, but based off of this, they're not generalizable to things outside the data set, or we cannot assume that they will be generalizable to things outside the data set. And then the things that have the least bias have a more meaningful AUC because the AUC is not based off of memorization because the k nearest neighbors is not high. And then another thing to note when looking at the breakdown of the active and inactive biases is that often the active set gets a negative number, and then the decoy set, instead of trying to be optimized to 0 itself, cancels it out, making the data set potentially harder to classify and not bringing both values down to 0, which would make a better model or have less bias. And so things that we have done to look at this problem and do things a little bit differently is, one, we changed this bias score so that it wouldn't have a negative component and there would be no canceling out. And another thing we did was try a weighted metric. So instead of optimizing splits based off of a bias score, the contribution to the AUC or whatever metric you're using is not as strong when your test drug is very close to something that is in the training set. And so here's some results of looking at these different methods. In this graph, the blue shows splits that were made just randomly. And then in the orange, and so each dot is a different protein in the data set. And then the orange is with splits that were done by the original bias score. And then the green is the new bias score that does not allow for canceling out. And so the x-axis is our bias, and the y-axis is our something that we came up with for this study called nearest neighbor similarity. So this is the similarity with 0 least similar, 1 the most similar, of having the same results as a nearest neighbor result with k equals 1 as a measure of generalizability. And with that, that means that the lower the score, the less it's like the nearest neighbor model, and potentially more generalizable it could be. And so what we can see here is that our new score in green is better at creating splits with the potential for generalizability with this bigger spread of the nearest neighbor similarity, and it also produces more scores, more consistent scores. And in this random, it looks like everything is perfect, but we have no way of knowing if this is because it is only memorizing the training data because it's all practically the same as the nearest neighbor model. So these could potentially be making models that cannot generalize and make new discoveries. And here we show our models with the weighted metric. And so the blue is the splits with the original bias score, and the orange is again the splits with our new bias score, and the green is random splits, but the PRAUC is based off of a weighted metric. So we can see here that the new bias, our orange score, still creates models that have poor performance. And this is because we're limiting the training data for the models by optimizing them to not have any testing data that's similar to training data. The blue with the original bias score, most of them are under 0.5 PRAUC. With our orange, we do get some above it, but still most of them are below it. But then with our green, we have most of them are above 0.5. And the reason for this is that we're not limiting our training. Any data can be training data, but then the contribution to the PRAUC is downweighted if it's very close to something in the training. So we're not limiting what we can learn from, but we're having a more reliable PRAUC that can produce better models with no data wrangling. And so to get back to the original question on the presentation is, can machine learning discover new drugs? And so the takeaway from this is that if care is not given to creating generalizable models and looking at the data that goes in them, they're never going to be able to use for new discoveries. So I think that there's still a lot of unlocked potential, but we have to make care to look at the data and think about whether or not they can be generalizable and not just, a study of any application in machine learning. And so here's just some acknowledgments of people I've worked with over the years in this area, from different national labs that I've visited, and here at the University of Kentucky with our Center for Clinical and Translational Sciences and the Markey Cancer Center. And with that, I'm finished, and if there's any questions.
Thank you so much, Sally. That's a very wonderful presentation. While we're waiting for questions coming, I have one question. So for the feature input data, the features of input data, how big is the feature data set?
So yeah, there's lots of different ways to featurize the data sets, and I think some of the initial machine learning studies that what was happening is there's the feature space is too large, and there's patterns that can be found in anything if the feature space is large enough. So there's software that creates cheminformatics features that you can just quickly generate thousands of features for each drug. Protein features, there's so many different ways to look at it, combinations of amino acids, the structure. So the feature space is huge. So really figuring out the right way to limit it is one of the problems.
The second question would be, from your own research or from the literature you reviewed, what do you think the most important features for example, drug discovery in here is like potential bonding, and are they all equally important, or some features tend to be superior than the other?
I think that maybe creating new ways of inputting three-dimensional structures and really encoding, what's happening on a physical and chemical level is going to be important to make generalizable models and not particular features that can just be quickly generated with informatics type software, and that we're really still working on that.
Awesome. And the next question would be, do you think in the future machine learning model will replace white lab workers, white lab researchers?
No, I think that even if we really advance the state of the art in machine learning and drug discovery, that everything's always going to need to be experimentally validated and better understood, because biology is extremely complex, and there's just so many variables that we don't even understand when it comes to what's happening in our body when we take chemical compounds.
The next question would be, what is your favorite algorithm for bounding prediction? And have you tried any ensemble approach? And if you did, what do you think the results should be look like?
I missed the second part of that.
Oh, have you tried any ensemble approach, like combine multiple models together? And if not, what do you think the results would be?
So the most experience I have is actually, before looking at the machine learning side of things and using molecular docking, and the software that I used was Vina. It's just what I have the most experience in. There are quick calculations. And when we were doing that, we were looking at ensembles. So that all had to do with three-dimensional structures of the proteins and the drugs. And typically in molecular docking, the protein structure is kept static, but we know that in our body, proteins aren't static in crystal structures, but they move around. So what we did was molecular dynamics of the proteins, which is basically a movie of how proteins could move around using Newton's laws of motions. And then you take this movie and extract some snapshots of hopefully significant conformations of the protein, and then do the sufficient calculation to these different structures. And then we did an ensemble approach where we combined the scores from several structures, and we found from the benchmark data set we were getting better results doing this over using crystal structures.
Awesome. And the next question would be, when you are doing the machine learning or any statistical learning, machine learning, deep learning approaches, there should be always a normalization to normalize the features. However, from a lot of this physical chemistry experiment, the values or the data collected range very differently. Do you have any insight on how to normalize these features?
I don't know the correct answer of what's best to do, but I think the most important thing when you're looking at it is to always make sure that you're not changing the meaning of the data by doing it.
Okay. So what is the acceptable level of accuracy for the drug development, or from your proposed method? What is the acceptable levels that you can tell this data or this model will be able to predict very sustainably usable?
So are you asking what?
The level accuracy, in order to achieve we can have it like machine learning to be able to use it for the drug discovery.
Yeah, I think that still is going to be dependent on quantifying the generalizability of models as opposed to just having toy problems kind of where it's all benchmark data and having good performance for that. It's an expensive, calculation or it's an expensive experiment. So it's hard to take a computational method where you do a lot of predictions and really compare it to experiment because, the cost involved with it. But I just think that moving forward, just care always has to be given into whether or not models generalize to things outside of the training data, because it's not going to lead to new discoveries if not.
And the next question would be, is there any disease dependence when you are implemented your research? For example, in the gene, in the targeting gene product, is there any specific subgroup this kind of disease or this kind of genes to analyze the results instead of trying to analyze all the gene products at once?
Yeah, so we have done some modeling where we used multiple proteins that were all kinases because they're common drug targets in cancer research. And, sometimes different kinase inhibitors look really alike. So building models that are, specific, but can also tell which kinase it will bind to and which it won't would be important. So we have looked at that some. But I really think that there was a lot of, issues with data overfitting still. And really it was that research that prompted me to start looking at the data sets in, more detail themselves.
Awesome. I think that's all our questions. We do not, I didn't see any more questions. Yes. So I think thank you so much, Sally, for your wonderful presentation. And YouTube, if you're watching this YouTube, you're more than welcome to contact with Sally or connect with me to ask him more questions. I will forward to Sally, and let's keep it going. Thank you so much. And remember, coming back, this is this machine learning in biology stream from the AISC. Thank you so much.
Thank you.
Thank you.